My name's Martin Sheldon, and I'm going to talk to you today about protecting the bovine endometrium against damage caused by pathogenic bacteria. By the end of the talk, I hope that you'll have a better sense and be better able to answer these four questions. How important is postpartum uterine disease in dairy cows? How do cows prevent uterine disease? How do pathogenic bacteria damage the endometrium in cows? And what are the mechanisms providing tolerance to pathogens in dairy cows? Uterine disease is a problem for modern dairy cattle. So if we think 70 years ago, the dairy cow was a different breed. It produced far less milk, used far less energy for milk production, and uterine disease was quite uncommon. Figures are difficult to attain, but probably less than 9% of animals developed any form of uterine disease. The modern dairy cow, however, commonly develops disease. About 40% of animals have some form of uterine disease. And this is in parallel with a big expansion in milk yield and a big expansion in the energy demand for milk production. Interestingly, the academic research and literature has followed this trend, so that 40, 50, 60 years ago, there were very little research on postpartum uterine disease. But since 2000, there's been a rapid expansion in the number of papers published on PubMed discussing or reporting about uterine disease in cows. Uterine disease is a costly problem for modern dairy cattle. Metritis costs farmers up to $410 per case. This is a cow on the left here with metritis, with a discharge from the uterus, which you can see behind her. Clinical endometritis reduces first service conception rates, increases days open, and increases culling. And the presence of pus here in these samples of purulent vaginal discharge reflecting clinical endometritis is typical. And the total cost for treatment, lost milk production, and replacing infertile animals is about $2 billion per year between the USA and the EU. Uterine disease is common, as I say. So if we think of the percentage of cows on the vertical axis here and the first 70 days postpartum on the horizontal axis, all cows are infected with bacteria, and these are culture samples. So these are cows with abundant bacteria shortly after calving. And then there is a decline over time. But 20 to 40% of animals have some form of metritis, and then about 20% of animals have clinical endometritis. Beyond that, there's a whole bunch of animals here, maybe 20, 30, 40% of animals that have subclinical endometritis, and relatively few to begin with a normal endometrium. And even 60 days after parturition, not all the animals have a normal endometrium. In fact, the endometrium is inflamed and damaged when there is clinical endometritis or metritis. So in image A, this is the endometrium, a section of the endometrium with a single columnar epithelium and an underlying stroma. In image B, which is an expansion of the section, you can better see the single columnar epithelium and the stromal cells. There's areas of damage, and you can see migrating neutrophils here and here, indicating inflammation in the endometrium. Not only does the uterine disease affect fertility at the time that the purulent discharge is there, but there are also long-term effects on the reproductive tract. So this is a study we did some years ago where we induced endometritis in cows by infusing two of the most common pathogens, Escherichia coli and Trupperella pyogenes. So we synchronized these animals, we infused E. coli and T. pyogenes together, or a control culture broth. The animals developed clinical endometritis when infused with the bacteria. And then we collected oocytes, endometrium and fallopian tube and granulosa cells either 60 or 90 days later. And these samples were used for RNA-seq analysis to look for differentially expressed genes, which were then analysed by ingenuity pathway analysis. And the image on the right are changes in gene networks 2 to 3 months after the initial infusion of bacteria for each of the tissues. And you don't need to read the list of networks down the side. In fact, these are truncated here, just to know that these are cell death, cell injury, inflammation networks, for example. And where there's a change, you can see a different color. White is no change. And you can see that the endometrium, caruncle and intercaruncular, the fallopian tube, the isthmus and the impala, and the ovarian follicle, granulosa and oocytes all have changes in genes and their networks 2 to 3 months after the infection. So this is a long-term detrimental effect on the reproductive tract. So postpartum uterine disease is important, but how do cows prevent uterine disease? Because not all of them develop disease. Well, the first point to make is that the endometrium is always infected with multiple species of bacteria after calving, whether that's gram-negative E. coli, as I've already shown you, or gram-negative anaerobes like Prevotella fusobacterium, or gram-positive bacteria like Trupperella pyogenes, which we infused in the previous slide, and Staphylococcus aureus. So the endometrium is always infected. And in this molecular study, on day 0 after parturition, animals that remain healthy or subsequently develop metritis actually have a similar landscape, a similar microbiome on day 0. And it's only several days later, when disease develops, there is a divergence in the microbiome. So they all have a microbiome, they all have pathogens in the uterus after parturition, and there's not that much difference between those animals that are healthy at the start or will develop metritis. So the key question is, how does that happen? How do some animals develop disease, and how do resilient cows prevent postpartum uterine disease? Well, resilient cows prevent postpartum uterine problems by avoiding, tolerating, and resisting the pathogenic bacteria. So these are the 3 strategies that organisms can use to avoid disease caused by pathogens. Whether that's bacteria, viruses, or fungi, it's the same principle. It's a general principle. So avoidance, here in green, is about limiting the exposure to bacteria, hiding from the bacteria, behaviors to avoid diseased animals, for example, or maintaining their hygiene, as another example. So this is about avoidance. Resistance, in the red, is what we commonly study, and this is fighting the infection. This is limiting the number of pathogenic bacteria, killing bacteria, using the innate and adaptive immune system, and stimulating inflammation to clear and eliminate the pathogens. Tolerance is a relatively new feature of preventing disease. It's widely studied in plants, but only more recently studied in animals. And this is about limiting damage caused by pathogenic bacteria or excessive inflammation. It's not about killing the pathogens, it's about tolerating their presence. So it's about protecting tissues against toxins and repairing damage. Now, if we think about these 3 components in dairy cows, avoidance is very difficult. If you're an extensive system, beef cows on a mountainside, that's fine. You can avoid pathogens, you can avoid other herd members. But in most dairy herds, intensive farm production means that the cows find it difficult to avoid other animals and avoid pathogens. Of course, we can help them avoid trauma to the reproductive tract, which is the biggest risk factor for uterine disease. So the odds ratio for endometritis is listed here for the major risk factors, particularly retained placenta, which 34 times as many animals will have endometritis if they have retained placenta compared with animals that don't. Stillbirth, twins, assisted calving, first calving, all these are associated with increased trauma to the reproductive tract. So we can help by reducing trauma, but of course the animals cannot avoid this trauma per se, so it's not an intrinsic behavior. So we're left with resistance and tolerance. Now what we aim for in this cartoon, if calving is here on the horizontal axis, and this is the first 28 days postpartum, we want both resistance on the top and tolerance on the lower panel. And what we aim for is a rapid, robust inflammatory response to the pathogens that always are present in the uterus, control of the pathogens, and then active resolution of inflammation to a homeostatic healthy status. Similarly with damage, control the damage, limit the damage, repair the damage, and return to a healthy status. And that's what resilient dairy cows do. But unfortunately, a lot of modern dairy cows, whether this is associated with metabolic stress, which we've talked about elsewhere, or tissue trauma, there's often an inadequate or a slow inflammatory response leading to chronic inflammation, and there is often excessive tissue damage and impaired repair mechanisms leading to chronic pathology and disease. So compromised resistance or tolerance to pathogens causes postpartum uterine disease. Now, in terms of protective mechanisms for resistance and tolerance, the epithelial barrier that I showed you the histological section of, here's the single columnar epithelium with tight junctions and a mucus layer. This forms a substantial barrier to infection in the uterus and helps tolerate the presence of these bacterial pathogens. However, as you know, in the postpartum uterus associated with the trauma of parturition and the physiological changes of tissue turnover, often the epithelium is lost, which exposes the underlying sensitive stroma to these bacteria and their virulence factors. There are other things like antimicrobial peptides and acute phase proteins that can also help limit the damage that these bacteria cause. But in the postpartum uterus, there is exposure of the stroma to these pathogens. So in that case, cows rely heavily on innate immunity. So innate immunity to the pathogens, whether that's gram-negative bacteria, which have lipopolysaccharide, LPS, as their major virulence factor, or gram-positive bacteria that have, for example, lipopeptides. And these are recognized by innate immune receptors, things like toll-like receptor 4 or toll-like receptor 2, on epithelial and stromal and immune cells that lead to the production of cytokines like interleukin 1 and interleukin 6, and chemokines like interleukin 8, which have the role of drawing in neutrophils, which help in the inflammatory process to clear the pathogens. There's also damage caused by pore-forming toxins, which we'll talk in detail about shortly, and this leads to epithelial damage and stromal damage, which in turn leads to the release of damage-associated molecules, things like interleukin 1 alpha. And these are sensed by the innate immune system as well to augment or scale the inflammatory response to meet the challenge faced by the pathogens and the tissue damage. So innate immunity is a central feature for resisting the pathogens, killing the pathogens, eliminating the pathogens, controlling the pathogens in the uterus. And this combination of pathogen and damage associated molecular patterns stimulates this innate immune response and inflammation through these innate immune receptors and production of damage associated molecules like interleukin 1 alpha, which leads to an augmented response. So the main section of my talk now is about bacteria causing damage to the endometrium and how they protect the endometrial cells against this damage. So the first question is, how do pathogenic bacteria damage the endometrium? Well, I'm going to focus initially on Trupella pyogenes because this is the pathogen most associated with the severity of uterine disease, and its production of a pore-forming toxin is prototypical of many of the other pathogens that produce toxins like E. coli or Staphylococcus aureus. So Trupella pyogenes is a gram-positive. It's actually a commensal in the uterus and the skin, and the respiratory tract, but it becomes pathogenic in the postpartum uterus, particularly when the epithelium is damaged and the underlying stroma is exposed. And as I say, this is the pathogen most associated with the severity of uterine disease, particularly the presence of pus. T. pyogenes secretes a cholesterol-dependent cytolysin called pyolysin. The gene is called PLO. And this pyolysin forms pores in cell membranes. So this is a pore-forming toxin. And this damages cells. And you can actually see that if you put T. pyogenes on a blood agar plate, you can see where the bacteria has been streaked here, that the blood has undergone hemolysis, and you only see the red of the blood agar plate where there is no streaking of bacteria. And just to verify this, if you make a PLO deletion mutant, in other words, a bacteria that doesn't secrete pyolysin, then you can see the streaking, but you can't see hemolysis because there's no pore-forming toxin. And this is also true in epithelial endometrial cells and stromal endometrial cells. So if you look at cell viability, the first bar is the control, 100% survival, 100% viability. The second bar is an interesting control. This is LPS, and LPS in concentrations that are found in the uterus does not cause cell damage. However, Trupella pyogenes, when applied, causes almost complete loss of cell viability. But of course, the pyolysin mutant that doesn't secrete pyolysin, the cells survive. And this indicates that this pyolysin is a major virulence factor for this bacteria. So how do cholesterol-dependent cytolysins work? How do they damage the membrane? Well, the bacteria secretes soluble monomers of this protein. These monomers bind to the plasma membrane of cells, whether that's epithelial cells, stromal cells, or immune cells, where they aggregate and oligomerize and then extend their beta sheets to form a pore or a hole in the plasma membrane. And if you look at the cartoon on the right, you can see the hole here, and this allows the leakage of potassium ions and proteins. The protein that most people measure is lactate dehydrogenase because it's easy to measure, and because it's abundant inside cells and not outside cells. And these pores are in these cholesterol-rich areas of the plasma membrane. If there's sufficient leakage of ions and proteins and other cytosolic contents, then there is cell lysis, cytolysis, and cell death. So if we just think a little bit more about this damage to endometrial cells, it's not only the leakage of ions and proteins, there's also disruption of cell barriers, alteration of the cytoskeleton, and cell lysis. So in this cartoon on the left, here are two normal epithelial cells with their nuclei, proteins in their cytosol. Here's the plasma membrane joined by tight junctions to form a barrier. And here's the actin cytoskeleton that maintains cell shape. And if you have damage with bacterial toxins, as I've just shown you, but also by cell stress or excess inflammation, then you get plasma membrane disruption or blebbing of the plasma membrane. You get leakage of ions and proteins. And as I say, LDH, lactate dehydrogenase, is the protein people usually measure. The nucleus is misshapen, the barrier has broken down, and the actin fibers are disrupted and dissolved away so that they are less distinct. And then you get plasma membrane rupture, cell lysis, and cell death. And what is useful is you can measure several of these factors. And I'm going to show you 3 of them today, but we can measure many others. But these 3 give you a general picture of the cell damage caused by pore-forming toxins like pyolysine from Trupperella pargenes. So on the left, we have LDH leakage. This is leakage out of the cell into cell supernatants. And the LDH leakage is on the vertical axis. On the horizontal axis is either a control challenge or challenge with increasing amounts of pyolysine. And as you challenge with more and more pyolysine, you get more and more leakage of LDH from the cells. At the same time, if you measure cell viability, so this is MTT assays, these are assays that measure the ability of cells to convert a chemical to a color substance, and it's a measure of viability. Here's the control. The cells are all viable, and then as you add increasing amounts of pyolysine, you get reduced cell viability with cell death. Then on the right are images of the actin cytoskeleton, which is in white. The nucleus is in red. So here are some control cells, nice nuclei, nice striated cytoskeleton of actin, and nice clear cell boundaries and cell shapes. However, if you challenge with pyolysine, the arrows point to blebbing or protrusions of the plasma membrane, misshaped plasma membrane, and then you've lost much of this striation. You just have a grey, granular look, and you don't have the striation, and the cell shape is disrupted. So this leads to the question, if that's what causes damage to the pathogenic bacteria, what are the mechanisms providing tolerance to pathogens? We already talked about the epithelial barrier, although this is lost in the postpartum uterus. But if we think about our main pore-forming toxin, pyolysine, it is a cholesterol-dependent cytolysin. And so one of the thoughts is, can animals regulate cholesterol, or can we regulate cholesterol in plasma membranes to protect against these pore-forming toxins? And I'm not going to spend too much detail on the molecular aspects of the regulation of plasma membrane cholesterol, but just to make the point that most cellular cholesterol, 90% or more, is in the plasma membrane. And this cholesterol is obtained from LDL cholesterol in the bloodstream through the LDL receptor, or from cholesterol biosynthesis via the mevalonate pathway. And then there is regulation of the amount of cholesterol, either by the formation of cholesterol esters or by sensing of cholesterol in the endoplasmic reticulum that then regulates cholesterol synthesis and cholesterol efflux, or by degradation of cholesterol or oxidation to oxysterols, which also regulate cholesterol efflux and cholesterol biosynthesis and other pathways, as I'll show you later. So we know a lot about cholesterol plasma membrane regulation, but can we alter this to protect cells? Well, an interesting thing if you measure cellular cholesterol is that stromal cells, which are more sensitive to pyrolysin, as it turns out, than epithelial cells, also have more cholesterol in their plasma membrane than epithelial cells. But we can actually reduce cellular cholesterol with this chemical called methyl beta cyclodextrin. It's a large chemical and it sucks up cholesterol sitting in a pocket in the molecule. And if you take stromal cells and you treat them with methyl beta cyclodextrin, you can reduce cellular cholesterol to the concentrations found in epithelial cells. And indeed, that is protective. So if we look at cell viability here, here's the control, all the cells are viable. Here's killing with pyrolysin in the absence of methyl beta cyclodextrin. But if you treat the cells with increasing amounts of methyl beta cyclodextrin for 24 hours before the pyrolysin challenge, at the higher concentrations of pyrolysin, you completely protect against, at the higher concentrations of methyl beta cyclodextrin, you completely protect against pyrolysin-induced damage. So that's one way we can protect the cells. Another way we thought about protecting the cells is thinking about cholesterol biosynthesis. So here's our pyrolysin pore in a cholesterol-rich domain in the plasma membrane. And as I said earlier, some of that cholesterol is derived from cholesterol biosynthesis via the mevalonate pathway, which includes things like statins that block HMGCR, that makes mevalonate. And you can also block other sections of this biosynthesis pathway, including blocking FDFT1, which is squalene synthase here. So inhibiting squalene synthase also protects the endometrial cells against damage. And one of the inhibitors of FDFT1 or squalene synthase is zarrogosic acid. So here's some evidence of its protective ability from a few years ago. So here's our LDH measurement again. This is the control challenge. This is the pyrolysin challenge in the absence of zarrogosic acid. But if you treat the cells with zarrogosic acid for 24 hours, then the pyrolysin-induced LDH leakage is reduced. In terms of cell viability, here's the control. Here's killing with pyrolysin. But if you treat with zarrogosic acid, the higher concentrations protect against that pyrolysin-induced cell death. And then if you look at the actin cytoskeleton, these images show cells damaged with pyrolysin. So they've been treated with vehicle, challenged with pyrolysin. The cytoskeleton is not very clear. The nuclei are misshapen. The cells are blebbed. But if you treat with zarrogosic acid and then challenge with pyrolysin, you still see some changes in cell shape, but the nucleus is much more clear and the cell outline is more clear. There are fewer blebs. So those are things that we can do to protect cells against cell damage. And that led us to think, well, are there things that the endometrium does or the cow does that are intrinsic mechanisms that protect against cell damage caused by pore-forming toxins? So rather than extrinsic treatments, are there intrinsic protection mechanisms? And so we started looking at oxysterols. Now we go back to our cartoon of cholesterol regulation, As I pointed out, cholesterol can be oxidized to oxysterols, and oxysterols either stimulate cholesterol efflux, block the cholesterol biosynthesis pathway, or increase cholesterol esterification to reduce cholesterol in the plasma membrane. So this is a feedback mechanism. And what's interesting is that oxysterols are abundant in animals. And there are many of them. There are oxidized forms of cholesterol. So here's cholesterol, and here's a series of enzymes in red that oxidize cholesterol to different oxysterols. And the two that we're interested in are 27 hydroxycholesterol. It has a 20 hydroxy on the 27 carbon here, and 25 hydroxycholesterol, which has a carbon with a hydroxy here. And then there are a whole stream of downstream metabolites formed from these oxidized forms of cholesterol. And what's important is these are oxysterols that are present in ovarian and uterine fluid. So if we collect follicular fluid from dominant ovarian follicles or from growing ovarian follicles, or we collect uterine fluid, and then we do mass spectrometry, which is rather difficult to do for oxysterols, but we are fortunate to have a collaborator in our building who is one of the world's experts. If you measure 27 hydroxycholesterol or 25 hydroxycholesterol, you can find nanogram per mil amounts of 27 and 25 hydroxycholesterol and metabolites, which I'm not showing, in the uterine and ovarian fluid. So they are in the reproductive tract. And then if you take endometrial epithelial cells and challenge them with lipopolysaccharide, the virulence factor from E. coli that stimulates innate immunity, or pyolysine from Trupella pyogenes that forms pores, then you get increased abundance of 25 hydroxycholesterol in nanogram amounts, whether you measure the 25 hydroxycholesterol per se, or whether you look at the downstream metabolite 7-alpha 25 dihydroxycholesterol. So we then explored whether these oxysterols alter the protection of endometrial stromal cells and epithelial cells against pyolysine. So I'm just showing the stromal cells here, but you get the same effect in the epithelial cells. So if we look at LDH leakage, here's the vehicle with a control challenge, no leakage. Here's a large amount of leakage when the vehicle treatment is challenged with pyolysine. But if you treat with increasing amounts of 27 hydroxycholesterol for 8 to 24 hours at the nanogram per mil amounts, you reduce LDH leakage. Here's cell viability, here's the control. Excuse me. Here's the control. Here's pyolysine killing the cells, and then treatment with nanogram per mil amounts of 27 hydroxycholesterol protects. And you see this with the actin cytoskeleton. Here's pyolysine damaging cells treated with vehicle with these blebs, misshapen, loss of striation. And here's 27 hydroxycholesterol treated cells, better shape, fewer blebs, and more evidence of the cytoskeleton. So that's 27 hydroxycholesterol. 25 hydroxycholesterol is as effective. These are epithelial cells in this case, but it's the same for stromal cells. Here's our LDH leakage. Here's the vehicle challenged with control, no leakage. Then the vehicle challenged with pyolysine, large amount of leakage, but then this leakage is reduced by nanogram amounts of 25 hydroxycholesterol. Here's cell viability, the control challenge, and then killing by pyolysine, but treatment with nanogram per mil amounts of pyolysine protects against damage. The same with the actin cytoskeleton. Here's the vehicle pyolysine challenged cells that are damaged, misshapen, blebbed, And here are the treated cells with 25-hydroxycholesterol and challenged with pyrolysin that maintain their shape and maintain the actin cytoskeleton. So these oxysterols protect against this cholesterol-dependent cytolysin. But we also wondered if there was a wider, more generalized effect on pore-forming toxins. And so we used Staphylococcus aureus alpha hemolysin, which isn't cholesterol-dependent. However, to our surprise and excitement, we also found similar results. So here's LDH, here's the vehicle-treated cells, challenged with a control, no leakage. Challenge the vehicle treatment with alpha hemolysin and you get leakage. But if you treat with 27-hydroxycholesterol, then you reduce that leakage. Here's cell viability. So here's our control challenge in the vehicle treatment. You challenge with alpha hemolysin and you kill most of the cells. But if you treat with 27-hydroxycholesterol, you prevent that cell killing by alpha hemolysin. In the images, again, you can see these cells are misshapen when treated with vehicle, but challenged with alpha hemolysin. On the other hand, when you treat with 27-hydroxycholesterol and then challenge with alpha hemolysin, you can see cell shape is better maintained and you can see the actin cytoskeleton. How does it work? Well, I'm not going to show you all the detail, but essentially these oxysterols reduce plasma membrane cholesterol, as in the cartoon, and you can quantify that by the pyrolysin binding. So, as I said, pyrolysin binds to cholesterol and depends on cholesterol. And so if you treat cells with pyrolysin, you can then wash the cells and apply an anti-pyrolysin antibody to detect the pyrolysin binding to the cells. So on the left are 3 controls. So these have a control challenge after treatment with vehicle, 27-hydroxycholesterol, or the cholesterol depleting agent that I showed you earlier, methyl beta cyclodextrin, and there's no pyrolysin binding. Same 3 treatments followed by pyrolysin challenge. In the vehicle treatment, as you'd expect, when pyrolysin is applied and then you probe for pyrolysin, you get a lot of binding because these cells are killed by the pores. But if you treat with 27-hydroxycholesterol or methyl beta cyclodextrin before the pyrolysin challenge, you have a reduction in pyrolysin binding. The alpha tubulin is just a loading control. And then this is quantified below, and you can see the pyrolysin binding in the vehicle treatment, but the reduction in pyrolysin binding, which reflects the reduction in plasma membrane cholesterol in the cells treated with 27-hydroxycholesterol or the control methyl beta cyclodextrin. And so, excuse me, in summary, oxysterols alter pyrolysin accessible cholesterol in the plasma membrane. They reduce it to protect the endometrial cells or to allow the endometrial cells to intrinsically protect themselves against pore-forming toxins. So whether these oxysterols come from the ovary or from uterine fluid, and whether that's in response to pyrolysin and lipopolysaccharide from bacteria, the end effect is that these oxysterols help protect against damage caused by pyrolysin. So in summary, my answer to the 4 questions are that postpartum uterine disease is common, costly, and has long-term effects in modern dairy cows. However, it's not inevitable, so resilient cows prevent uterine disease by avoiding, tolerating, and resisting pathogenic bacteria. Pathogenic bacteria secrete pore-forming toxins that damage endometrial cells and cause tissue pathology, and being able to tolerate this tissue damage is a key protection mechanism against uterine disease. And in the final section of the talk, I've given you an example of how either reducing cellular cholesterol with agents like statins, methyl beta cyclodextrin, or squalene synthase inhibitors can protect cells. But actually, more excitingly, there is an intrinsic protection mechanism in animals that uses oxysterols to protect endometrial cells against pore-forming toxin damage. And so I'm going to leave you with the final slide that makes the key point of much of our recent work, and that is that resilient cows prevent postpartum uterine problems by avoiding, tolerating, and resisting pathogenic bacteria.